Is welcome again of participants or friends and colleagues. I think it's the second webinar of the WESTPAC OA webinar series today. I think today we will have a Dr. Li Yang Chang. He will be moderator for this this webinar. I would like to give the floor to the Dr. Li Yang. Li Yang, please, you have the floor.
Hear me?
Yeah.
Okay. So yeah, thank you for this chance that I can be the moderator to serve. And today the first speaker is Dr. Anita Eutrophic from Xiamen University. She is a PhD candidate in marine chemistry, and her research interest is carbon and nitrogen biogeochemistry, dynamic under the influence of eutrophication, hypoxia, and ocean acidification in the marginal and estuary systems. So her topic of today's talk is integrative effects of the oxygenation and acidification along the sub-estuaries in Upper Gulf of Thailand. So welcome, please, Anita.
Hello, everyone. Thank you so much. I'm glad to be here, and I'm going to share my screen now.
Yeah, you can share screen now.
Okay, everyone see my screen? Hello.
We can hear you.
Oh, thank you. Okay, I'm going to talk now. Hello, everyone again, and I'm glad to be here to share my research study. My name is Kanita Uthaipan, a PhD student at Xiamen University, and my today topic to talk, that is the integrative aspect of the deoxygenation and ocean acidification.
Your screen is not
Yeah,
your screen. We could not see your screen yet.
Cannot see your presentation PPT, please.
Oh, it's ongoing now?
Yep.
Okay, thank you.
There you go.
Okay, this is my topic again. Integrative effect of the deoxygenation and acidification in the Upper Gulf of Thailand. And this is my outline today. So we are going to the first topic, estuary and coastal zone are land and sea interaction zone that play a significant role in the human livelihood and are strongly affected by human activity. Large amount of inorganic and organic nutrients drain into this area. The enormous nutrient input can lead to eutrophication, and when combined with the radiation driven by freshwater input, stratification water cause a severe decluttering of the production and respiration. Production organic matter, while oxygen at the bottom water. During oxygen organic matter respiration will reduce pH concentration and raise PCO2 in the bottom water, and also decrease the saturation of the carbonate aragonite. This respiration at the bottom play a role in the vertical and also horizontal gradient in the carbonate parameter. The respiration at the bottom right, the increase dissolved inorganic carbon, PCO2 level, and also lower pH and DO at the bottom, plus water acidification at the subsurface water in the coastal zone. And currently, population in Thailand rising to 69 million people. At the same time, Thailand is experiencing and direct effect of the climate change at worldwide, such as more flooding and drought frequency and other water resources issue do occur. Water pollution issue have become more stress pressure in Thailand due to deteriorate of the watershed for the agricultural development. Industrial population rapidly grows in 1965 and sharply increased in the 1985. The first record hazard for the pollution in Thailand occurred in the 1970s, namely the molesty incident, caused by the spill of the molesty released into the Maeklong River. This resulted the fish line eliminated in the river and the cocoa covering several thousand acres in the upper Gulf of Thailand. While the period of symptoms of the eutrophication and hypoxia began in the developing country about the 2000 and the developed country about 1960. And in here, anthropogenic nutrient and carbon are hugely important aspects of the marine environment change in Thailand. First, water quality over the watershed basin and coastal zone degraded, especially the water quality in the estuary and coastal in the upper Gulf of Thailand due to the low oxygen concentration and high nutrient pollutant. Secondly, that is the phytoplankton bloom also varied both sides, not yet only in the Gulf of Thailand, but also in the Andaman Sea. And lastly, we found the hypoxic condition or even not low oxygen concentration also have been reported in the upper Gulf of Thailand, Bandon Bay, Pak Panang Bay, and also Songkhla Lake in the southern part. And the upper Gulf of Thailand is a core economy and biodiversity area in Thailand. Bigger picture show the location of the upper Gulf of Thailand with the coral reef, seagrass, seashell farm, and also important marine species, for example, chod mackerel or in Thai called plateau, and marine mammal. Those are economically important species or cultural organisms. They reside in response to ocean deoxygenation and ocean acidification and other strata in coastal and estuary water. In the future, there may weaken ecosystem resiliency, threaten the survival of marine organisms. So we pay attention in this area and aim to investigate hypoxia and ocean acidification in the upper Gulf of Thailand. The ocean deoxygenation and ocean acidification have been reported in upper Gulf of Thailand. The upper Gulf of Thailand is a concerning area because here currently it combines effects of the oxygen deficient and water acidified had potential more immediate effect on the health of the estuary, nearshore ecosystem, economy, fishery resources, and also social use. For example, lot in the shellfish harvest and harvesting and coral reef health. And my significant of this, firstly, global database show that coral reefs are associated with the more of the half of the no tropical depth zone worldwide, with a greater than 10% of the coral reef at an elevated risk to the hypoxia based on the location and also global risk factors. Currently, the tropical coral reef mortality is caused by hypoxia, but data had been unreported. Secondly, from the predicted scenario of the coral under the increasing of the carbon dioxide at the 375 ppm, coral reef still held, but if the carbon dioxide increase concentration and plus 2 degrees, seaweed will be more covered and dominated at the 500 ppm. And in the worst case of the over 500 ppm and plus 3 degrees Celsius, coral reef will disappear and be replaced by clumping masses of the eroding coral skeleton. And recently, Viyakan study in 2015 indicated pH level caused by the substantial decline in lava settlement rate in the experiment at the 7.6 and 7.9. All lava were unable to complete metamorphosis, and metamorphosis delay was observed for the coral reef species Posilophora damiconic. So we will investigate the changing pH, aragonite saturation state for the hypoxic consumption assumption, and examine how carbon dioxide emission under IPCC representative concentration pathway will change in the future carbonate system in the Upper Gulf of Thailand. To get better understand of the hypoxia and ocean acidification in the Upper Gulf of Thailand, we collect data of the carbonate system from the three parts. From under the collaboration of Thai-China Joint Laboratory for Climate Change and Marine Ecosystem, include hypoxia project established under the sub-project of the MUMSI in 2019, hypoxia project data and ocean acidification project from the Department of Marine and Coastal Resources since 2016 and 2020. This is showing in the big figure. And here we observe the carbonate species and also involve parameters such as the DO concentration, and the variation of the river discharge also shown in here, where in Thailand they have the two seasonal monsoon. The variation of the water discharge that started from the southwest monsoon bring the moisture and precipitation into Thailand, caused of the increasing of the runoff, and start in May and peak in the October. This period is probably to form the hypoxic condition close to the coastal zone. And finally, we will start with our result. I would like to introduce you that is the Lysi. Lysi, that is the triangle, the area here, and Eastern Island is a small island in the eastern side of the Upper Gulf of Thailand. And this green triangle, that is the data for the river estuary panel, and the blue dot, that is come from the entire of the cruise conducted in the Upper Gulf of Thailand. Firstly, we will start with the temperature at the depth, at all the depth, the mean at all the depth, the mean temperature of entire water column was 30 degrees Celsius, and the difference between northeast monsoon and southwest monsoon is just only about 2.1. Salinity ranged from 2, and significant variation influenced by the river discharge. Generally, salinity fluctuation found at the estuary and coastal zone. Salinity at the Lysi is quite steep variation expected during the high, except early the high discharge into the Upper Gulf of Thailand, and the potential temperature related to salinity that show there was a seasonal variation between the northeast monsoon and southwest monsoon. High salinity show the different characteristics of a water mass due to the temperature changes in this area. And for the DO saturation in each island and also Lysi show the variation range between 190 to 200 above. Why the high variation it occur in the estuary and river zone close to the coastal zone due to the phytoplankton productivity, especially during the southwest monsoon. And the seasonal variation a little bit appear in the Lysi and Eastern Island, but both of this area is a low impact from the river. For the PCO2, water quite all in the Light Sea and Eastern Island, but in the contrast, the estuary zone it shows lovely variation. We found the highest value almost 30,000 in Meklong River in river. So the seasonal variation show the low concentration during the southwest monsoon because of the photosynthesis and high concentration in the northeast monsoon due to the mixing process. And the parameter we are going into that is concept and high related. So firstly, we start at the Light Sea and Eastern Island. We will see just only consistency variation, fluctuation, and a little bit peak when have a runoff river. For example, in the August in 2018 and 2020, and ranges between the southeast monsoon and northeast monsoon mean water 7.76 and 7.95 of the entire of the upper Gulf of Thailand. So generally in the ocean, pH concentration will be almost 8 or 8.1, but entire of the Gulf of Thailand, we have the data that is 7.9, almost 8. And aragonite also show in the same trend, and the value is between the southwest and northeast monsoon stand at the 2.4 and 2.7 of the entire column. And here, this figure is come from the 2019, and this is from the my collaborative team that distribute for the current water. And here the figure show that it's the bottom and surface water current what in the different world in the different direction. The buoyant water exhibited in the northwest corner, we can observe from the salinity and the range of the velocity between 0.02 to 0.1 meter per second. The tidal cycle was a mixed tidal during investigated time, and water masses was well mixed in the eastern zone of the upper Gulf, and stratification was occur in this area nearby the estuarine. And here DO, pH, and aragonite, we did show the extremely related both of the surface and in the bottom water. But however, decoupling processes between the productivity and respiration processes was not clear because during investigative time that quite low river discharge, but we still observe the trend of the decoupling process. And this one, we're looking for the temporal change of the pH and aragonite from the Light Sea because there are longer time series data we have. This data series show the water the identical property in terms of the salinity and temperature. Here we still observe the influences of the river discharge or fresh water in southwest monsoon season or during the rainy season, such as in June 2018 and July in 2019, and which play a significant role in the pH and aragonite saturation variable. The time series pH institute indicate that the slightly decrease of the pH value during the southwest monsoon and seem to be the decrease over the time. At the below figure, we can see much more blue color compared to the upper panel. So in here, we found the minimum value pH that is 7.6 and mean value at the 7.9 of all the data collection. Here, aragonite is quite same with the pH concentration. Besides, the DO concentration was low near the seabed, and so why we found the high pH concentration in some area during the southwest monsoon. And this is the result. We go into the impact of the micro respiration play a role in the carbonate system. The result indicate the eutrophication and acidification in the upper Gulf of Thailand. We will assess the impact of respiration play a role pH and carbonate, and we assume the organic matter decomposition is the same as with the refuse stoichiometry equation. And this figure show the decoupling process between surface layer and bottom layer the carbonate species, both of the surface and bottom water. And here we modeled the pH and aragonite from the reconstruction data TA, DIC, and using the AOU data, and determine DIC will added in the surface water and also subsurface water. And the time we will investigate that the three terms. Firstly, it's pre-industrial, present, and in the future. And here is the result analysis of the all data collection. A correlation between the pH and DO concentration. Line C and offshore station show the high value of the DO and pH in the contrast. Low of the oxygen and pH was found in the station near the estuary, and also found the some of the hypoxic condition. A moderate line, three line represent the comparison of the different amplitude of the carbon dioxide level, presenting the condition of the pH and oxygen depleted with the hypothesis of the organic matter respiration. Firstly, in the pre-industrial, when the carbon dioxide level was 2,800 ppm, the saturation concentration indicated pH value started from the 8.09 and will decrease 0.36 degree along the respiration till the zero salinity of the DO. At the present, in 2019, when the carbon dioxide level was 410, and today from the Malau station is 417, and the value pH in here show the currently pH 7.98 and will decrease to 7.52 in at the zero salinity of the zero concentration of DO. And in the future, when the carbon dioxide rise to 800 ppm, pH value will sharply decrease. If we compare the pre-industrial to present value pH compared to future, the decrease of the pH that is almost 0.5 unit from the hypoxia condition. This result will more significant pH decrease caused by the atmospheric and anthropogenic carbon dioxide input to the oxygen depleted and carbon dioxide enriched. In here we call oxygen depleted and carbon dioxide enriched. But if in the high saturation of oxygen, we call the oxygen enriched and carbon dioxide depleted. And in here, we can see the depleted in the upper Gulf of Thailand from the pre-industrial to present will decrease 0.1 and decrease 0.21 when the oxygen deficient. If compared to the North Gulf of Mexico, a little bit different, just only even below, and the effect of the oxygen rich droplet to carbon dioxide pool is defined as the enhancement of ocean acidification emphasizing the biological amplification of the initially defined ocean acidification concept. This you can follow from the paper from Chai in 2011. And in the future scenario, at the end of this century, the buffering capacity of water subjected to eutrophication induced hypoxia will be reduced further, result in the overall of the dropped 0.81 unit from the present oxygenated to the oxygen deficient in the future in this area. And the same, I also investigate aragonite concentration. Ocean acidification is causing a decline in saturation state of the calcium carbonate minerals such as aragonite. This is more soluble than calcite. Aragonite is a building block of the stony corals that are the primary framework of the habitat builder of the coral reef ecosystem. Decline in aragonite saturation state result in the decline in calcification rate of the coral reef. This study is showing the projected result of the greatest decline in aragonite saturation state by the organic matter respiration and ocean acidification. Here, the decline of the aragonite started dropping about from the pre-industrial to the future scenario. Currently, the value that is 2.7 of the entire and 3.1 at the station nearby the coral reef. So this scenario may alarming investigated affected drop of the pH and aragonite saturation state caused by the respiration will treat to the vulnerability of the coral reef in this zone. Vidyakan and et al. In 2015 reported that drop of the pH at the 7.9 and 7.6 will decline in the development of the data lava state. In here, my result also show drop of the pH in the future that will decrease aragonite about 1.13 unit. Does it mean the coral possibly cannot building their skeleton because of the decline of the aragonite together with the pH value? And in here, a representative concentration pathway, RCP, in the greenhouse gases concentration trajectory adopted by the IPCC. Therefore, pathways are consistent with the wide range of the possible chain of the carbon dioxide in the future. This study calculated that the predicted of the pH aragonite saturation due to the organic carbon remineralization with different level of the carbon dioxide in the atmosphere provided by the IPCC since 2014. The model of the pH result show the representative of the my value decreased from the RCP 2.6 from the 0.2 unit will drop to the 0.29 unit when the RCP at the 8.5, and in the same time aragonite also decreases and from the 2.6 to 8.5 that is the 0.09 unit to the 1.26 unit. In the summary, the pH and aragonite will drop it 0.92 and 1.26 unit in the next century in the upper Gulf of Thailand. And the lastly of my conclusion, thirdly, eutrophication may increase the susceptibility of the water to ocean acidification in the upper Gulf of Thailand, in which ocean deoxygenation and ocean acidification is coupled, that driven by the anthropogenic carbon and also climate variable on the temperature and precipitation that mentioned in the water mass characteristics and biogeochemical chemical processes. And secondly, The time series data indicate that pH and aragonite saturation undergo two drastic reaching in the droplet of the pH institute temperature and aragonite approximately about 0.1.3 units. In determine and in the near future I have to do. Firstly, the effect of the temperature increase because of the saturation need the temperature function. And secondly, the long monitoring data, the target site study are emphasized. And thirdly, that is a monitoring station hypoxia and ocean acidification are still challenging in this area because the upper Thailand, even this area small compared to other site study, but the capacity building and when we have the crew in Thailand, my suggestion and comment from my result, firstly, the carbonate calculation may have the capacity to understand of the mechanism of the coral metabolism processes and also can predict the future reef health status, which require to continue a more prolonged monitor observation both of the carbonate system and also other parameters that involve in the eutrophication and ocean deoxygenation in the Thai water, not yet only in the upper Gulf, the Thai water is essential. Lastly, my result will be benefit for the biologists working on the effects of the ocean acidification in the upper Gulf of Thailand. This value from my result may help you to decide on the setting of the experiment. And this is my future plan. I am still young and I am still have a dream. And this is the entire of the my future plan. Hopefully I will have a target study site in the seashell and fish farm, also in the sea glass and coral reef that involve to the eutrophication, hypoxia, ocean acidification. These three things are involved together if we want to understand the impact of the anthropogenic carbon play role in this area. So we have to do it together. And this is my work. Since I was young, I have dreamed in 2010 and at that time, quite a long time and we don't have the capacity to do this. And my dream it come true when 2019 under the support of my advisor and Thai-China collaboration coming with the first hypoxia project for me. And I hopefully in the future, in near coming, in 2025, I can involve these teaching together and have some result. And here I don't want to waiting until 2100 because maybe my life is short, but I hopefully at that time our earth is safe now. So lastly, I'm looking for the kind of support from the senior, from the expertise, not yet only in Thailand, but also international supported. And hopefully I will have the collaborative teamwork in the next future. And lastly thing, I have to say the million thanks to all the person involved in my research. I cannot do it without of them. So I appreciate all of the person involved in this project. And our team do appreciate for your attention for my speaking. Thank you.
Thank you, Kate.
Mine.
Okay, thank you, Kate, for your nice presentation. And we will have our Q&A section after three speakers present their work. So the next speaker is Professor Maria Lauda Santiago Magro. She is from Marine Science Institute, University of Philippines, and her research interest is nutrient biogeochemistry and coastal systems and sediment water interaction and aquaculture, marine culture, environment impact of the agriculture, marine agriculture, blue carbon, and so on, and ocean acidification, of course. So, her topic of this presentation is coastal acidification and other eutrophication problem in mariculture area. So welcome, Professor Maria.
Good afternoon, everyone. My name is Malu Maclone, and I would like to talk to you today about our experience in the Philippines on coastal acidification, which we are seeing as the other eutrophication problem in our mariculture areas. The content of this talk are as follows. I will give a background on how mariculture is important in the Philippines, how mariculture in our study site has caused eutrophication, algal blooms, and fish kills, and then discuss an emerging impact of mariculture, which is coastal acidification. Then end up with some take-home message. I would like to start off by showing the pH distribution map of the Philippines, which is actually a work in progress. This map was inspired by the first WESTPAC workshop on research and monitoring of the ecological impacts of OA on coral reef ecosystems, held in Phuket, Thailand in January 2015. There were 4 other workshops that followed this first one. To date, we have data from 59 locations all over the country, with many of the samples collected by other researchers from the respective study sites. What we do is we send them our OA kits that include bottles, preservative, and everything they will need to collect the sample, plus instructions on what to do and where to collect the samples. After collection, they send the samples back to us. Now let us zoom in to the upper part of the map and we see red dots. The symbol red means low pH level, and in the scale it's less than pH 7.9. The red dots are in the mariculture areas of a town called Bolinao and its neighboring town called Anda, which are located in the northwestern part of the Philippines. The question to ask is, why is pH in these areas low? How is low pH related to mariculture? Mariculture or fish farming in the coastal and marine environment falls under aquaculture. This figure shows us that aquaculture contributes over 50% to total fish production in the Philippines. This means that fish is needed to supply human consumption, which is 40 kilograms per year or 109 grams per day. There is an increasing trend towards mariculture in the country because there is no more space for brackish water and freshwater aquaculture. Also, mangrove conversion to aquaculture ponds is no longer allowed. Moreover, mariculture is being promoted as a livelihood diversification option for coastal communities in the Philippines. In this map, we see the location of where the Philippine government plans to establish mariculture parks and zones, and they are all over the country. And among the species cultured, milkfish is the second most important next to seaweeds. Here again is a map showing Bolinao, and this town is a major supplier of milkfish in the country. Bolinao and Anda are neighbors. These 2 towns share common waters. They both engage in mariculture of milkfish or chanus in fish cages and fish pens, and unfortunately they both experience algal blooms and fish kills. The question is, why is this happening in these 2 towns? Now let us examine the contributing factors to describe what is happening to mariculture in Bolinao and Anda. What started out as a small-scale operation in the 1990s has now expanded to a major industry in the 2 towns. On the left panel is a map from the Coastal Development Plan for Bolinao, established in the late 1990s. The dark green rectangles are symbols for fish pens and cages and where they should be located. The carrying capacity or prescribed limit for the number of pens and cages is 544. On the right is a map showing the fish pens and cages in 2007. You will notice that there are more fish structures here compared to the map on the left. What is a consequence of so many fish structures? In a study we did, we measured the water velocity inside and outside a fish pen on the left graph and a fish cage on the right. Blue bars are water velocity for inside and red bars for outside. What we found out is that velocity of water passing through a fish pen is reduced by 66% and 35% when passing through a fish cage. So can you imagine how much water flow is reduced if you have many of these structures side by side in just one particular area? For Bolinao, the number of fish structures did not remain within the carrying capacity of 544 units, but it actually doubled in 2002. And that is when the first massive fish kill happened in the area that resulted in big economic loss of around $8 million. Since then, the town has kept the number of fish structures to within the carrying capacity up to this day. However, algal blooms and fish kills continue to occur. With expansion in fish structures, you create eutrophic conditions. This is a diagram to illustrate what is happening to the feeds given to fish inside a fish cage. Ideally, every kilo of feed should be converted to a kilo of fish. Hence, no feed is wasted. However, the practice in Bolinao is 2 kilos of feed are given to produce a kilo of fish, or a feed conversion ratio of 2. This is because the owner desires to produce bigger fish. As a result, 1 kilo of feed is uneaten or wasted, and so the carbon, nitrogen, and phosphorus in the feeds dissolve in the water column or settle as particulate material to the sediments. Hence, the source of water quality problems in mariculture areas are the uneaten feeds and fish waste products. The feeds currently used in Bolinao-Anda are not good quality feeds. They easily disintegrate in a few minutes, as we saw in our experiment, hence the nutrients are readily released to the water column. These graphs show the release of nutrients from feed material. In an experiment done at the lab, feeds release as much as 379 micromoles of ammonium per day, which translates to 850% increase in ammonium concentration in 4 days. The same trend is seen for the other nutrients. Hence, when you look at the time series of nutrient concentrations in Bolinao waters, even if the number of structures is maintained within the carrying capacity, nutrient concentrations remained elevated, as well as chlorophyll values, making the environment eutrophic. The effect of eutrophication is also seen in the sediments. When you examine the sediments under a fish cage, what you see is a core on the left, where sediments are black, sulfidic, and with a white mat of sulfur-loving vegetative on top. Organic carbon content is high at 14%. For comparison, the sediment core away from the fish cage is shown on the right. It is brown, oxygenated, with burrows where bioturbators live, and with only 1 to 2% organic carbon content. Because of eutrophic conditions, we now see algal blooms happening. During the massive fish kill in 2002, the bloom that caused brown coloration was from a plankton called Prorocentrum minimum, which comprised 99% of the dinoflagellates in the water column during that time. This is a timeline to show the frequent occurrence of algal blooms in Bolinao and its surrounding waters over the years. And to this day, blooms still occur. We have seen blooms change every month. For instance, in May, it was Alexandrium, which is a toxic species that predominated. And in the following month of June, it was Skeletonema, a non-toxic species. And this has been going on for years with various algae blooming. The map on the left are locations of where algal blooms of Pyridinium, Alexandrium, and Gymnodinium have occurred all over the country. And on the right are locations of mariculture areas around the Philippines. The question is, do we see a correlation? The experience in Bolinao-Anda can be replicated in these mariculture areas all over the country, which is something we do not want to happen. And when the bloom dies, there's decomposition that uses up oxygen. And with no oxygen, fish will die, resulting to fish kills. In an experiment to examine the change in oxygen due to decomposition of feeds, we found out that DO levels can be completely depleted in 24 hours, as shown in the top graph. And with no oxygen, fish will die. During the massive fish kill in 2002, not only the milkfish died, but also reef fishes, as seen in the picture at the center. Here is a newspaper report of another recent fish kill in Bolinao and Anda. This is a map of areas affected by fish kills and associated algal blooms in the country. You will notice that the most number of occurrences is in Bolinao and Anda, which is seen as number 1 in the map. Fish kills are costly. This is a trend line of the economic losses due to fish kills over the years in Bolinao, even if the carrying capacity is maintained to 544 structures. To summarize, the consequences or environmental impacts of mariculture in Bolinao are eutrophic waters, algal blooms, both toxic and non-toxic, oxygen loss, and fish kills. Now, one other impact that has emerged is coastal acidification. The other impact, which is damage to ecosystems or coral reef resources, will be discussed by my colleague Patrick Baitan next week, May 19, in this OA webinar series. The impact on ecosystems is quite relevant because when you look at this image of Bolinao and Anda, you will see the presence of pens and cages in red symbols, together with corals in pink symbols and seagrass in green. So the marine resources in these habitats can be affected by eutrophic and acidic waters coming from the mariculture areas. The influence of water from mariculture areas, marked as X in the figure, is seen in this hydrodynamic model for Bolinao and Anda, where you see effluent from mariculture areas in brown reaching the seagrass and coral areas during the wet season. This influence, however, is not seen or is not as extensive during the dry season. The presence of pens and cages in Bolinao and Anda, which you see in the map as brown dots for cages and green dots for pens, have caused nutrient and organic loading. From the reaction below, the decomposition of organic matter would release nutrients that make waters eutrophic and carbon dioxide, which causes coastal acidification. Hence, what is happening is reflected in the spatial distribution of nutrients in Bolinao. The top panels are for nitrate and the bottom for phosphate. The dots represent nutrient concentration with bigger dots for higher concentration and smaller dots for lower concentration. Nutrients in corals are in blue, seagrass in green, and mariculture area in red. The biggest dots are in the mariculture area since this is where nutrient concentrations are highest and conditions are eutrophic. In this figure, the circles or dots represent pH values. The smaller the circles, the lower the pH. From the previous slide, the mariculture area is where nutrients are higher. This is also where circles are smaller, which means pH values here are lower. On the left is another plot of pH distribution in Bolinao-Anda. What is alarming is that pH values have gone down to pH 7.7, which we see in the time series on the upper right. pH 7.7 is quite acidic compared to oceanic pH of around 8.2. Moreover, the pH we are now seeing in Bolinao mimics the low pH predicted for the end of the century or in 2100. Hence, monitoring pH is important to determine short-term trends as we see in the 2-day time series, or longer trends as shown in the 3-year time series of pH to examine the longer-term effects of mariculture in Bolinao waters. Using a pH sensor, we have seen the short-term pH variability in some of our sites in the pH map, as shown in the insets that I will describe in the succeeding slides. A pH time series tells us the short-term diurnal variability, as we see here in 3 sites. It also tells us episodes of low pH, as we see in the time series for 2 mariculture areas, or a highly variable pH as seen in bubble vent sites. With information on pH and other carbonate parameters, we can get insights on the community metabolism in a reef area. For instance, as shown in the diagram on the left, the slope of total alkalinity and DIC will suggest whether there is net calcification or net production. This type of analysis was done by my MS student for a reef flat in Bolinao. He examined the seasonality of the slope. He saw lower slopes during the wet season, which suggests that more production is occurring over calcification. And this is attributed to the strong influence of mariculture waters on the reef flat during the wet season. To conclude, these are the learnings from our work that I want to leave with you. 1. The significant environmental impact of mariculture is associated with the release of waste from uneaten feeds and waste products. These environmental impacts are eutrophic waters, algal blooms, fish kills. 2. Coastal acidification has become the other eutrophication problem in mariculture areas. 3. There is a need for mariculture to be sustainable because of the plan for mariculture expansion in the Philippines. Recommendations to make mariculture sustainable include proper site selection and spacing, establishing the carrying capacity for mariculture sites, use of good feeds, and improving on feeding practice. 4. Coastal acidification should be monitored. And 5. The impact of acidification will be seen in coastal areas. Hence, there is a need to do more ocean acidification research. It is imperative for us to do ocean acidification research in the Philippines because the Climate Change Act, which came up with the National Climate Change Action Plan, did not include the threat of ocean acidification. It just focused on increasing temperature and sea level rise. It is important to include ocean acidification because it will have an effect on food security as well as ecological and environmental stability. That would be all. Thank you very much for listening.
Okay, thank you. Thank you for your presentation. And now is our last speaker, Professor Aline Tan. She is director of Center of Marine Coastal Study of USM, and her study interests is marine culture, marine ecology, biodiversity, and conservation. And the topic is effects of ocean acidification on oyster aquaculture. Please, Aline.
Okay, thank you, Liang. Thank you for the introduction. I would like to take this opportunity to thank IOC-WESTPAC, Dr. Somkiat, the head of OA studies under IOC-WESTPAC, and also Mr. Wingzi Zhu for giving us the opportunity to share our work on OA in this webinar series. The title of my talk today is Effects of OA on Coastal Aquaculture. The scientists involved in this research are myself, Nitya, Nohanis, Abby Wu, and also Prof. Zulfiqar Yassin. I will present my talk in the very layman terms as possible in order to be able to reach out to a broader audience of different field of research. Okay, most of us know that increased CO2 is related to ocean acidification and would cause biophysical effects such as effects on the calciferous animals like mollusks, crustacean, corals, echinoderms, and some phytoplankton. And it has implications for fisheries and aquaculture, like what has been presented by Dr. Malo. And then it has potentially reduced production for calciferous marine resources and ecologically related species and decline in use, which has been proven quite clearly in the earlier talk. Currently, the current knowledge on ocean acidification in terms of aquaculture is we have good laboratory experiment, however, limited number of field studies in the low pH environment. If you look at this slide, it shows that if CO2 continues to rise unchecked, computer models show that acidification will deplete carbonate ions in much of the ocean by year 2100. Like what been also mentioned by the earlier presenters, turning the waters corrosive for many shell-building animals. And if you look here, why is ocean acidification important to be studied under the platform of IOC-WESTPAC? Because if you look at this information being displayed by National Geographic, it seems that there is not much information being shared on the global platform in our region. And then I would like to say also, ocean acidification is not a new research area, and OA research has been initiated many years ago. It was been talked about many years ago. However, there is still knowledge gaps in Southeast Asia, especially the effects of OA on aquaculture, because there's lack of field-based experiment and also lack of comparative experiment, and also with the complication of multi-stressors. The general public, we have to admit that the general public do not take OA seriously as compared to marine litter or plastic pollutions or even coral bleaching. The public can see and feel deeply for the impacts of plastic pollutions when they see many plastics in the stomach of seabirds, or dolphins being strangled by plastic fishing lines, or our corals turning colorless during bleaching. But what about the impacts of OA? It is mainly the changes in water chemistry, and these changes has not been linked visibly to something the general public could understand or worry about immediately. Like just now, Prof. Malo was mentioning, but about fish kills, which is related to ocean acidification. But a lot of time, the public would link the fish kills with hypoxia or eutrophication rather than OA itself. And many times also, scientists do not relate OA impacts to our daily lives or convert the impacts into economic values, such as OA's impact on aquaculture. So with this, despite the lack of attention and commitment from the public as well as the policy makers, the impacts of OA cannot be denied and taken lightly by all of us. Dr. Mala has just presented a talk on effect of OA and hypoxia on fish aquaculture. Mine will be focusing more on bivalve aquaculture, which is experiments on oyster mainly. The tagline here is we are losing our bivalves. Malaysia is losing our cockles. We have been producing cockles at 100,000 tons per year, and since maybe 8 years ago, our tonnage, our landing tonnage of cockles has dropped 86%. That is a huge loss in our economy, and that is also a trigger, a threat to our food security, right? So that is why when we know that is impact, why are we losing our bivalve? Why are we losing our shellfish? The organisms that has shells, because due to low pH is usually related to ocean acidification, high temperature, because when you're talking about climate change, a lot of times the public would link it with increase in temperature. And also, for the case in Southeast Asia and also in Malaysia, is mainly on the low salinity, because like what been presented, we are actually talking more on coastal acidification, where freshwater input has been playing a more significant role in changing the environment. And when these are all synergistic stresses in the Southeast Asia, and it is actually very, very difficult to talk about just one stresses, in most of the research. And then the question here is, can they survive in the tropics? So with this, from Thailand, presented by the first speaker, to Philippines, now I bring your attention to the scenario in Malaysia. In order to study the effect of ocean acidification in the tropics, 2 sites were chosen. The focus area is on the Straits of Malacca, where there is 2 sites, Site A and Site B. These are the description of both the sites. Site A is about 1 kilometer from the coastline near an island. That means it's quite a distance away from freshwater influence. And it has a mean pH of 8.3 in average. And the salinity is 30.3 parts per thousand, ranging from 28 to 32 parts per thousand. That means even though heavy rain would not actually bring down the salinity in this area. And then the other site is Site B, which is along the Merbok River. It is located in the river system, which is subject to huge water runoff during raining season. And this area been exposed to mean pH of 7.29 because of the influence from the fresh water. And the mean salinity is 20.5 parts per thousand, with a range of 7.25 parts per thousand. What we do is we use hatchery produced oyster seeds, meaning that we expose the oyster seeds to both the site and using 2 species, which is the Chrysoistra iridale and Chrysoistra belgeri. Both of these species are the commercial commercial oyster species being used in Malaysia. So these oysters I've been exposed to 2 different sites, and the parameters measure is the survival, the growth, and feasibility to reproduce. Yeah, and this is the quick result of the adult oysters. Yeah, by putting the oysters in the different sites. Yeah, site A is the full strength marine site, and the site B is the estuarine low pH site. And we can see that both CI and CI is meaning Quassia evidently, CB is Quassia belteri. You can see that the survival for both the species are relatively high in both the sites. Yeah, these are the adults. Yeah, these are the adults are both relatively high in both the site, and the growth is basically faster. Yeah, faster in site A compared to site B. And the shell thickness, yeah, for site A, which is the marine water, with normal pH, is usually thicker compared to the site which is lower in pH. Yeah, you can see oysters at site B showed higher survival. However, there's no significant difference compared to site A. This is the stage where we are talking about adults. Yeah, so both Quassia belteri and Quassia evidently shows that they can actually tolerate acidic environment when being exposed to different sites and being exposed to site B, which is lower in pH and considered as acidic environment. Yeah, but then if we look closely into the shell quality of the oysters, yeah, at different sites, this is the shell quality of oysters in site A. You can see that the shell is still nicely and thick, yeah, compared to the site B, yeah, which is lower in pH, which is acidic, and in layman term is caustic. That means it would dissolve the calcium. You can see that the shell of the oysters in this area, if we put it longer in this area, it would thin. It will become thinner. And once it's become thinner, it's more prone to predators. The crabs can easily attack them compared to the oysters in site A. Yeah. So in our next experiment, following to the earlier experiment, the broodstock, we are talking about the reproduction fecundity. So the oysters from site A and B were used as spawners. So the larvae produced were cultured at different pH level. So this is the results. Yeah, site A. Okay, bear in mind, site A is the full strength marine water with normal pH. Site B is the one which is in blue, which is one exposed to lower pH. Yeah, during the larval culture stage. You can see, just focus on the orange bar first, or the yellow bar. If you look at the yellow bar where the broodstock has been conditioned in the marine water with normal pH, which is 8.3, you can see that they are not able to survive. They are not able to survive at lower pH. Yeah, at lower pH. But then, however, for those broodstock that has been conditioned in site B with lower pH, you can see that they are able, they're able to survive all the way up to pH 7.2, even though the percentage of survival is much reduced. That shows that depending on the broodstock, if the broodstock is being acclimatized in lower pH, the offsprings has the tendency to survive at lower pH based on this simple experiment. Yeah. And if you look at the survival, yeah, the survival of the larvae from site B, there is no significant difference between pH 7.6 all the way to 8. That shows that they are able to tolerate, they are more hardy if they have been acclimatized much earlier in lower pH. Yeah. And in terms of the experiment on the effect of settlement, because just now is on reproduction and fecundity, and also the larval culture up to the pedivilager stage. This is from pedivilager stage for settlement. Yeah, you can see that in this graph, the stars are the area where for site A or for Belcheri, that we do not conduct experiment for site A because we cannot get the larvae to survive until settlement. So that's why I put a star there. That means there's no experiment being conducted, no settlement experiment being conducted. If you look at this experiment, the pedivilager larvae were exposed to different salinity upon settlement depending on species. Yeah, for Chrysospora eridali, the optimum species for settlement is 25. For Chrysospora belcheri, the optimum settlement salinity is 18. So during this experiment, we exposed the different species to their optimum salinity for settlement. If you look at the results, regardless of the pH of the pH being exposed, you can see larval settlement of broodstock from site A, yeah, was lower than site B as pH 7.8. Site A, remember the pH being acclimatized is 8.3. Even during settlement, it would not give us good settlement performance compared to the larvae from, or the broodstock from site B, which is exposed to lower pH. Yeah, and you can see that larval settlement of broodstock from site B show no significant differences between the pH 7.6 to 8. This is a reflection also if the settlement would not be affected by different pH level if the optimum salinity is given during the settlement stage. Yeah. As in conclusion, yeah, for both the species, Chrysospora eridali and Chrysospora belcheri, they are able to survive at sites with low pH level. However, the growth and thickness of shells are affected by the low pH level. But I would like to mention here also, which I do not present here, if we were to transplant back the oysters with thinner shells back to the pH of 8.3 or the normal marine pH, the shells are able to become thicker back to its original thickness compared to being brittle and thin. That means if the responses is irreversible, it can be returned back to normal. Yeah, so the larva tolerance towards lower pH level depends on the source of the broodstock. Is this acclimatization or is it adaptation? So more studies need to be conducted, because if it's adaptation, we have to look into the molecular level. Proteomics would be able to determine whether the adaptation or the tolerance is based on genetics, or is it just physical acclimatization over time. Yeah, the effect of pH would be insignificant if optimum salinity is provided during the larval settlement stage. Yeah, and then what I would like to emphasize here is the inherent adaptation. So inherent adaptation is something that it can be inherited, or is it already existing in the oysters itself? And based on the reference by Pauline Rose, she did mention that benthic organisms surviving in this current extreme area may display the positive carryover effect. Yeah, meaning that, you know, once they have been exposed, they can actually adapt. That means they can do adaptation and able to survive even though they have been exposed to lower salinity area. The reason why the site was chosen also on the Straits of Malacca, because in Malaysia, that is the highest aquaculture activities being conducted for Malaysia. That is where the aquaculture generates the money, the economy for the country. That is why it's important to study the changes of pH, the variation of pH in the Straits of Malacca. And then with this information, the question raised now is, will this species from this area, can it be a good target species for aquaculture in the Southeast Asia? If it can be adapted, if it can have a positive carryover effect, and it can acclimatize and be adaptable to low pH. Can the scientists now comes in together to actually have a species selection or selective breeding on the species that is tougher for aquaculture and make our aquaculture in the Southeast Asia sustainable, which has been mentioned by the earlier speaker. Our aquaculture need to be sustainable regardless of what the climate change is interfering. So by having the science intervention, we can actually come up with selection of the good target species for aquaculture to sustain and to secure our food security in the country and also in the region. So the increase of CO2 and ocean acidification, besides the biophysical effects, what we should need to know also on to look into more in-depth of the complicated responses. A lot of times we were saying that we need long-term monitoring of the situation of the OA, of eutrophication, but a lot of time we are just looking on the physical and chemical component of the whole issues of climate change. But then it's time also for us to link it with the biological, which is the biological component, which is giving us a more complicated responses, which is very much depending on the species, on the populations, or even on the different life stages. Like just now, when what I presented is on the different life stages, it gives us slightly different results. And then and many more other things that we need to look on the biological components, because from the biological components that will link us to our economic value of our food security and our economy. Yeah. With that, I would like to thank you all, and I'm open for discussion and comments. Yeah, thank you. And back to you, Liang.
Thank you, Aline. Before the Q&A section, and in case of we will forget about it, so we better take a photo, group photo before. So let's take a photo. Everyone, please turn your camera on. Happy face.
Oh, well, many of you haven't turned on camera.
Okay.
Okay, shall I start? Okay, done.
Okay. Okay, so now is the section for Q&A. So anyone have questions or anything? Oh, yeah, here we go. There's Asahiku. Am I right? Okay,
Yes.
here you go.
So that, yeah, I'm Masayoshi from Hokkaido University in Japan. So the, thank you for your presentations. And I have the tiny question to the Eileen. Yeah. So did you measure the omega argonite? Because
No, I did not measure the omega.
I really want you to do that, because to do so that you can assess the impacts by pH and also the salinity at the same time more clearly.
Okay, yeah, thank you for the question. And for your information, our team did not measure the omega. That is something that the next stage that we are looking into, the omega content as well as the molecular content, want to trace back the genetics, whether has it anything to do with the genetic responses towards environmental impacts. Yeah, thank you, Prof.
Yeah, thank you very much.
Wensi, you have question?
Oh yes, not question, but just something I think to seek your some kind of suggestion.
I think, you know, over the last 10 years, I think based on my experience in different groups, scientific aspects, I think nowadays, the way of developing science, the way of thinking about science, I think changed dramatically compared to the, you know, 10 years ago. So I think more and people emphasize the importance of integrated, right? Particularly in our region, integrated and also solution-oriented approach. And I think this is the positive moments that I can say. So the question is, for every study, for most of the presentation, I think on the last presentation, the last slides, we always say, oh, more research need to be done. More collaboration need to be made. Further investigation are necessary. I keep hearing, you know, this kind of description, but of course, I understand science developments require time, you know, intensive, extensive efforts over the time. My question is how to make that kind of things happen, right? For instance, take the, you know, the presentation provided by Eileen. So for that specific case, seems, you know, the physical team, chemical team need to work very closely with the biological team, particularly focus on their BioVa, right? How to make it happen, right? For the presentation provided by Milo, I think you emphasize on the side effects of the mariculture, massive mariculture in the environment. So coastal acidification actually is a kind of a result in this case, but of course, it also become a reason for another kind of environmental problem or you know, the productive problem. So I think how to initiate more meaningful scientific research rather than just leave the question over there. So this is something, you know, I think particularly, I believe you know this. I'm going to mention about UNTAC again. You know, whether we can really initiate some kind of concrete action, you know, based on the expectation everyone, you know, make on the last slide of the presentation, and also, you know, in consideration of their first speaker about the issue in the Gulf of Thailand. Okay, based on some you know, time series observation, we understand the change in the pH, in the eutrophication, and also in the saturation stage situation. And next, so what? Right, we also, as a scientific community, we also need to understand the concern of the decision maker, of other stakeholders. We always say we need long-term monitoring, time series. But this is always not possible, particularly in developing countries. So our research community need to find a way for ourselves, how to move forward. So this is something I have been thinking about this in order to find a way, solution, you know, for science development for this region, particularly for developing region. So this is my comments. Leon, floor to you.
Okay, thank you. Alan, you have a question? Or
yeah, I would like to add on a little bit to Bingzi's comment. I think he is very right in his comment, you know, because we have been doing a lot of science, and our science has not really translated to proper usage or to solution solving, which is something as a scientist, we now have to change our approach, not just doing science and to study more in depth. We have to now know how can our science benefits the society in that sense. And also to add on to our research on the oyster, on bivalves, you know, we are now actually, based on our results with that simple experiment, just by the survival and looking at the shell thickness and the growth itself. We now have been working closely with the coastal fishermen or coastal growers. Depending on the site, we will recommend them on what species to grow, and we collect the broodstock from their area to use as spawners, and we produce the seeds and return them the seeds. So the seeds itself are already having the inherent adaptation, that I've mentioned earlier, that they are able to survive even at the different various pH being exposed in that different sites. These are the small early steps that we are moving, trying to share our knowledge with the community and make them having a better yield and profit in terms of their aquaculture. Yeah, based on our simple findings. Yeah, I guess this is something all of us have to look into, and not just very much focus into the hard science. We have to now look into how to translate our science to useful component for everybody to understand. Thank you. Back to you, Liang.
Thank you. So, any other question, comments, and hello?
Yeah, maybe to respond to Wency's comment, I think it's important for us to bring science to policymakers, to local government leaders and environmental managers, because the way we're seeing it, the response is very selective. Like, for instance, they do not recognize ocean acidification, but they recognize the other climate change stressors. Then perhaps one question we should also look into on how we can solve or address is, how can WESTPAC have a regional influence on national issues? I mean, is there a way, because individually, probably as a collective group, we can have a stronger voice than individual basis.
Okay. So, any other questions or comments or suggestions or whatever?
Hi, Liang, Paul here.
Hi, Dr. Fu. Go ahead.
I think I also want to respond on Wency's comment just now. So Prof. Eileen had mentioned about involvement with industry or the local communities is important. And Prof. Maria mentioned about the government's involvement, especially policymakers' importance for the way forward, how we curb these OA problems. Perhaps one more angles we should look at is involvement of common people, like citizen science. I think Prof. Maria have done a good job in Philippines that involve different organizations doing this. I think this need to be enhanced. Some things that we know that doing OA in hard science requires spatial and temporal commitments. Perhaps it's time to, if like the WESTPAC can draft some kind of guidelines driven, like how we can engage the local people or common people, or we call it citizen scientists to involve in this. I think this will actually help to ease the pressure on the governments, like investment in terms of like equipment or resources on this long-term monitoring. I think this is all from my side. Thank you.
Okay, thank you, Paul. Any other question or comment? No.
Yeah, thank you, Liang. If you allow me, I just also, to share, because some thoughts, for our, our collective efforts in the near future, as I say, the very positive moment that I can say, particularly recently, is that I think for our research community, recognize very much the importance of bringing science to society. I mean, society including different kind of groups, community, policymaker, industry, business sector, NGO, those kind of things, even donor agencies as well. So ocean education, I think especially it's a long-term process, but we all know has a very long, far-reaching impact on ecosystem health, including mariculture as well, right? Food safety and security issues. So we have to, and also we face challenge how to sustain the ocean education in the long run. So this is something I think Melo say how the UN body can influence, that kind of national research agenda that can bring benefit to the country, many ways. But the first solution must be put forward from the research community. So this is what I said. Based on all the study we have over last 5 to 10 years, I see many ocean acidification research generate some result already, not only monitoring capacity, but also impact, impact on the mariculture, on the bivalve industry that we can say today clearly. First, for us, I think for our science research community, when you do the presentation, can you enlarge the scope of your talk? Putting more contents about the co-designed approach, not only focus on scientific results, but also put some content, how you think, how you design, the plan to address the issue through this co-design, co-involvement approach. This is very important. I think although I think we are interested in scientific result, but I think that part is also very important. It can give a very good story to different audience, right? Let them say how the research community considered the way to address the issue. And the second, good practice. The good practice here, it doesn't mean only we have very advanced monitoring capacity. I think this is far from enough. The good practice, how to use the science to address the socio-economic concerns. So if we have that kind of good story, build on that, we can produce a lot of materials, either in the form of video, right? Or in the form of the policy brief. So I think this is the very basic material that must come from our research community originally. But this may be overlooked in the past. So I think over the next 10 years, as I said, we really need to intensify our efforts in this part. So this is my suggestions. And then for us, IOC, we're board of IOC UN system, not enjoy very high status in the whole UN family, but we are doing our best, to highlight the importance of ocean research in different, political platform. So, but in the world depends on what material, right? We can use it to reach out policymaker. So this is something, preliminary thoughts in my mind, in terms of ocean education, of course, also including hypoxia, HAB, eutrophication, those issues, over the next 10 years. So this is my, preliminary thoughts at this stage. So I think in the future presentation, provide more, scope in your presentation about how you co-design the science, how you engage your stakeholder into the science development process, right? And come up how you use the science results to convince the relevant stakeholder. I think those parts are very important, right? Okay, Leon, thank you.
Okay. Thank you. Okay, so any other questions? No? Okay, so yeah, it seems there's no question then. That's it. Okay, so now here's the announcement for the next webinar. It will be held on May 19th next week at the same time. So Yeah, I think that's it, right? Yeah. So see you next time.
Thank you everyone. See you.
See you.
Thank you.
Hi.
See you. See you.
Thank you.
Very nice talk today, everyone. This is Apple.
Hi.
I miss you all. Bye-bye.
Thank you, speakers. Thank you so much.
Hi. This is Mr. Ali from Somalia. Remember my colleagues, Walalia University.
Thank you, everyone. Take care.
Take care, Eileen.
Take care, Malio.